Hello, my name is Eli Swart, and I am a field application scientist for Geneyx. And what I wanted to do is talk about some of the new features that we've integrated into version 5.7. So as many of you are familiar with, here I have displayed the variant dashboard. And the first thing that I wanted to do is address some of the new annotation sources that we've integrated into the software. So if I scroll to the right, under Effect and Prediction, we have the Splice AI. So at a high level, Splice AI predicts the probability of a variant being splice altering. And so really what this does is it predicts the splice site junctions from a pre-mRNA transcript, which enables precise prediction of non-coding genetic variants on cryptic splice sites. So this will ultimately allow you to look at splice site variants that occur in maybe intronic or deep intronic regions, and ultimately identify maybe previously overlooked non-coding genetic variants in the human genome for patients with rare diseases. So this can be added into your filter logic. You can also query, sort by ascending or descending, etc. Now, one of the additional annotation sources that has kind of been updated is the CAD scores. So CAD scores stands for Combined Annotation Dependent Depletion. And it ultimately assesses the deleteriousness of single nucleotide variants as well as insertions and deletions that span the entire human genome. And so we've integrated the CAD Fred score, which is kind of a scaled or normalized score, as well as the CAD raw scores. So if you want, you can add these into your filter logic. You can also query on them as well. So beyond these annotation sources, we've also integrated some nice intuitive features in the filter logic. One of the things that I wanted to point out is the ability to add or remove particular filters, especially ones that are kind of predefined. And so here in the filter logic, if you click on the plus icon, you'll now have the ability to add any of the predefined or pre-built genetic filter logics that we have applied, including those specific to genomic and genetic data, variant calling, Q&R, frequency, effect and prediction, as well as evidence. So if you want to add or remove those, we're giving you a nice dialog to do so. More importantly, though, in the filter logic is actually the weak association results. And so what this is really a non-clinical phenotyper. If you wanted to apply it to your particular workflow, you can select this option. It says, are you sure you want to also see weak association results? And ultimately what it's doing is it's going to add additional information from gene expression profile, mouse genome database, as well as large-scale gene publications. And so with more information, we're getting kind of a broader scope of the gene phenotype association. So it's ultimately going to impact your phenotypic score if you enter in a particular phenotype. But again, this is for a non-clinical. If you wanted to get kind of broader understanding of the gene phenotype association, this would be a great feature to integrate into your particular workflow. So beyond this, another new feature that we've integrated is in the copy number variants and structural variants tab. And so one of the requests by many of our users is to have a logo frequency database for the observed copy number variants across our cohorts. And so that's exactly what we've integrated. So now you'll notice we have an in-house number of local variants. So this will display the total number of samples that are present in your count, as well as if there is a similarly observed CMV event in that sample. So in this case, we have 9 hits. If we open this up, here we'll display the position of the previously observed CMV event, the sample it was observed in, the gender modifications, the zygosity, the analysis it was present in, as well as the phenotypes. And so at kind of a high level, that this can be really valuable in terms of eliminating or filtering out maybe common CMVs, or alternatively, if you're looking at a cohort for a rare disease, it might be valuable to see if the CMV event in one sample is observed in the other affected samples. So this really just kind of serves as a great resource for looking at the local allele frequencies for these copy number variants as well as structural variants. So beyond what we have displayed here, obviously there's a lot of new features, but one of the last things that I wanted to focus on is available in the actual analysis details panel. So again, the analysis details panel provides a great audit trail in terms of the protocol that was implemented. In particular, genetic models were applied, as well as phenotypes and gene panels, which you can see here. But what's new about this interface is actually this icon here, which allows you to set up a focused analysis. So the idea is if you have a whole genome sequencing or a large data file, you have the ability to perform a targeted approach. So let's say you wanted to focus on a particular gene, genes, or genomic regions. All you have to do is select this option and apply a new bed file, either an existing enrichment kit or a new target region. And what it'll do is it'll allow kind of a detailed investigation on the particular regions that you've entered, but it will also allow you to capture, maybe intronic variants such as regulatory elements, enhancers, those types of things. So it's a great resource. I would highly suggest playing around with it. It will create a new analysis, but it'll also allow for a more targeted approach for your NGS data. So again, this was a high-level overview of some of the main features that are available within Geneyx Analysis version 5.7. If you have any other questions about maybe some of the other features that we have integrated, please don't hesitate to reach out. You can always reach us at support@geneyx.com, and we will be happy to assist you with any of your questions. So with that, I wanted to thank you, and I'll talk to you later.